Mitosis is the separation of nuclear content. Its functions include the growth and repair of multicellular organisms, along with asexual reproduction, where the offspring receives exactly the same DNA instructions as the parent. Mitosis is important because each new daughter cell receives the same number and kind of chromosomes as the mother cell. The daughter cells are identical to each other in the mother cell.
The importance of meiosis, a type of cell division in which germ cells are produced through sexual reproduction, is the genetic variation. 3 events unique to meiosis are synapsis and crossing over, homologs on the metaphase plate, and separation of homologs.
The cell cycle. The first part of the cell cycle is the meiotic phase, and it includes mitosis and cytokinesis. The mitotic cell division begins with interphase, which is 90% of the cycle. Interphase is where the cell grows and replicates its chromosomes. It has 3 subunits, G1 phase, S phase, and G2 phase. G1 phase is where the cell grows. S phase, also known as synthesis phase, is where the chromosomes are replicated. G2 phase is where the growth is completed in preparation for cell division. The first phase is prophase. In prophase, the DNA recoils and the chromatin condenses into chromosomes, and the centrioles start moving toward the opposite ends of the nucleus. As the spindle fibers, also known as microtubules, begin to appear, the nuclear envelope disappears. The next phase is metaphase. In metaphase, the chromosomes line up along the middle of the cell with assistance from the spindle fibers that are attached to the centromere of the replicated chromosome. The third phase of mitosis is anaphase, and in anaphase, the sister chromatids become individual chromosomes and are pulled by the spindle fibers to opposite poles of the cell. The fourth phase of mitosis is telophase. In telophase, the chromosomes and other organelles divide into 2 portions as the nuclear envelope reappears around 2 daughter nuclei and the chromosomes revert back into chromatin. The final phase of mitosis is cytokinesis, which differs between animals and plant cells. Cytokinesis is when the daughter cells split into 2 individual separate cells. In animal cells, the cell pinches on both sides, forming a cleavage furrow which will move toward the center and split the cell into 2 daughter cells. In plant cells, the cell forms a plate from vesicles derived from the Golgi apparatus, but it starts in the center and moves toward the outer edges, splitting the parent cell into 2 daughter cells.
Prophase 1. In this stage, the DNA coils tightly and individual chromosomes become visible. Homologous chromosomes become closely associated in synapses and they exchange segments by crossing over. Metaphase 1. By this phase, the nuclear membrane has disappeared and the microtubules form a spindle. Spindle fibers attach to only one side of each centromere, and the 2 homologous chromosomes attach to microtubules orienting from opposite poles. Each pair of homologous line up on the metaphase plate. Anaphase 1. The microtubules of the spindle fiber shorten and pull the chromosomes toward the poles, taking both sister chromatids with them. Each pole ends up with a complete haploid set of chromosomes consisting of one of members of the homologous pairs. Telophase 1. The nuclear membrane reforms around the daughter nuclei. Each daughter nucleus contains 2 sister chromatids for each chromosome, attached to a common centromere. Because of crossing over, the 2 sister chromatids are not identical. In animal cells, a cleavage furrow forms. In plant cells, a cell plate forms. Prophase 2. The nuclear envelope breaks down and a new spindle forms. Chromosomes, each still composed of 2 chromatids associated at the centromere, move toward the metaphase 2 plate. Metaphase 2. Spindle fibers bind to both sides of the centromeres. The chromosomes are positioned on the metaphase plate as in mitosis. The kinetochores of sister chromatids are attached to microtubules extending from opposite poles. Anaphase 2. Breakdown of proteins holding the sister chromatids together at the centromere allows the chromatids to separate. The chromatids move toward opposite poles as individual chromosomes. Telophase 2. Nuclear envelopes reform around the sets of daughter chromosomes. The chromosomes begin decondensing and cytokinesis occurs. The mitotic division of one of the parent cells produces 4 daughter cells, each with a haploid set of unreplicated chromosomes. Each of the 4 daughter cells is genetically distinct from the other daughter cells and from the parent cell.
There are differences between mitosis and meiosis. In DNA replication in mitosis, it occurs in interphase before mitosis begins.
And in meiosis, it occurs during interphase before meiosis 1 begins.
Mitosis only has 1 division.
And meiosis has 2.
Synapsis does not occur in mitosis.
In meiosis, it occurs during prophase 1 along with crossing over between non-sister chromatids, resulting in chiasmata hold pairs together due to sister chromatid cohesion.
There are 2 daughter cells that are genetically identical to their parents.
In meiosis, there are 4 haploid daughter cells containing half as many chromosomes as the parent cell.
think about you, babe. I feel the same.